Good afternoon. I'm Mike Gilson. I'm co-director of the Center for Drug Discovery Innovation here at UC San Diego, and I'm a professor up in the School of Pharmacy, which you're sitting on the ground floor. And I'm really delighted to welcome you all to the second Drug Hunter Award lecture, which is sponsored and established by Wilson Sonsini, Goodrich, and Rosati. Wilson Sonsini, or WSGR, much too long, has provided legal services to some of the best known and most innovative venture-backed startups in the biotech industry, including local companies such as Abide Therapeutics, Vividion, Crinetics, Metacrin Therapeutics, and Fount Therapeutics. In the course of these activities, WSGR interacts with a lot of different scientists who are involved in developing new drugs, and they've now established the Drug Hunter Lecture and Award to recognize some of the most significant contributions to bringing new drugs to the market. Today we welcome Dr. Richard, is it Heyman? As the second recipient of this award, Dr. Heyman received his bachelor's in science at the University of Connecticut and did a Ph.D. in pharmacology at the University of Minnesota. Currently serves as the executive chairman of Metacrin and as a member of multiple other companies, and is also vice chairman of the board of trustees of the Salk Institute. So with that, I want to welcome Richard and welcome Mike Hostetler of WSGR, who will further introduce Richard and present the award. Welcome.
Yeah, thank you so much, Mike. Yeah, so I want to first thank the Drug Discovery Institute and everyone for co-sponsoring this. This is a really cool event that allows us to recognize some of the major contributors to the drug development industry. So what is a drug hunter? We call this the Drug Hunter Award. So what is a drug hunter? A drug hunter is a company, is a person who has, I would say, sort of a sixth sense for drug discovery. So it's not only just identifying opportunities, but it's then making those opportunities turn into drugs. And Rich Heyman is one of those people that has been able to accomplish that several times now. And so what we'll learn today about is some of the work that he's done that has led to a number of drugs that Rich has put onto the market. So very excited to talk about that and all that he's done in the drug discovery and drug hunting space. So one of the things I want to call out is, in addition to the award that we're going to be giving, WSGR is also giving up a $1,000 donation to Rich's favorite charity. And that charity is the Salk Institute and the Salk Education Outreach Program, which is a pretty cool program, actually. It supports reaching out to the local community and expose young students to science and STEM education initiatives. Perhaps actually finding the next Rich Heyman that's out there, we can hope. So without further ado, I'm sure everyone is interested in hearing what Rich has to say. So I would let us all welcome Rich, and I'd like to present him with the 2019 Drug Hunter Award. Thanks very much, Rich.
Well, thank you, Mike. Thanks to the Drug Discovery group here. And I'm really, really pleased to present some of the work that I've been doing here in San Diego for the last, I guess, 20 plus years, maybe 30 years. And it's actually great to see some people here who I both worked with, and actually one person in particular who was actually instrumental in some of the early work that I'm going to actually present today. So it's a real pleasure. What I'd like to maybe start with is to take a quick poll in the audience and just kind of go back a little over 100 years and ask you, what was the average lifespan for a woman back in 1918, if you can make a guesstimate? Somebody?
65. 45.
It was about 50, actually. And interestingly, if you use the 1918 term, it dipped down, and it dipped down both for men and women because of World War I, actually. But if you now fast forward 100 years, any guesstimate again for, let's just stick with women for now?
77.
84. 84. And the reason is, as we probably are all well aware, it's really the difference if you go back 100 years that many individuals were dying from acute diseases of viruses, diseases of bacterial infections, and actually just general hygiene and sanitation related issues. And if you fast forward 100 years, what we're really dealing with today is diseases that we often refer to as chronic diseases or diseases of the aging population. So we're dealing with cardiovascular disease, which is the #1 killer in the United States, cancer, Alzheimer's disease, and various aspects of obesity and diabetes. So as we think about drug discovery and drug hunting, we're really changing in terms of our mindset about thinking about treating patients that have chronic diseases. And so over the last 20, 25 years, that's really where I've been focused on, developing or trying to develop drugs that really are targeting aging diseases or chronic diseases. And of course, the simple solution, which many of you know, is you can change your diet, which can have a huge beneficial effect. You can exercise, which again can have a huge beneficial effect. But in general, maybe California is a little better than the rest of the country. Most people don't want to exercise more. Most people don't want to eat better. And so that's, of course, it's job security for the drug hunters, right? Because then you have to think about pharmacological intervention. And that's really where I'd like to focus today's discussion. Now, before I maybe jump into the actual research that I've been involved in, just Mike kind of gave a little of an intro, but maybe as many of us, when I started my career, I loved science. I got my undergraduate degree in chemistry on the East Coast. I moved to Minnesota to get my Ph.D., my wife as well, and always thought I'd stay in academia. And as we weathered 5 winters, you don't count it by years in Minnesota, you count it by winters, we decided with some arm twisting that we were going to move to the West Coast, and I was going to do my postdoctoral training just across the street at the Salk Institute. I had the honor and the privilege actually of working in Ron Evans' lab at a very, very exciting time. I joined Ron's lab in the mid to late 1980s. It was a very, very exciting time. And the tacit understanding that I had with my wife was that if we ever had any kids, they'd have to get a real job. Otherwise, she said, I'd probably be a postdoc there 30 years later. I just had such a good time. But the good news and the dilemma was my son was born in 1990, and I needed to get a real job. That was our deal. I thought I'd stay in academia, but I actually was quite fortunate that Ron had started a company in 1989 called Ligand Pharmaceuticals. It was focusing on nuclear receptors and transcriptional signaling. And I worked out a deal with both the Salk Institute and Ligand Pharmaceuticals where I could work 4 days a week at Ligand and I could spend 1 day a week still at the Salk. And I thought that was kind of a best of all worlds. You probably couldn't do that today with certain conflicts between industry and academia. But I was able to strike that deal, but literally within a few months, I was so darn busy at Ligand that I was there pretty much 24/7 as opposed to the Salk. But I'm fortunate enough that we continued lucrative collaborations, and I actually still have the pleasure of working with Ron on a particular project. So it's been an interesting opportunity. I've had the opportunity to work with multiple companies here in town, as I mentioned, starting with Ligand Pharmaceuticals, where we actually took technology out of Ron's lab, applying it to drug discovery. I kind of got the bug in 1999 to think about setting up my own independent company. We spun some technology out of Ligand and a few academic labs, and I helped start a company called Acceptor Therapeutics, focusing primarily in the area of metabolic diseases. And then in 2009, which is the majority of the story I'll tell you today, I got contacted by the former head of the National Cancer Institute, Rick Klausner, and 2 professors. One was Charles Sawyers, who was at Memorial Sloan Kettering at that point, and a chemist out of UCLA, Mike Jung. And they thought they had some new insights into potentially developing compounds for the treatment of prostate cancer. Would I get involved in maybe brainstorming about the idea of is there real legs in the context of that program? And then one thing led to another, where I basically helped write a business plan, started pitching, and as I'll tell you, we launched Aragon in the spring of 2009, working on hormone-dependent cancers. Also was very active, and I was the co-founder and CEO of a company called Saragon Pharmaceuticals. I'll also talk a little about that today. And we sold that company a few years ago, and since then I've been involved in a few startups locally here in San Diego, Metacrin and Vividion, which Mike mentioned, which is a chemistry company spun out of Scripps. I'm involved in a few Bay Area companies, and as I put in the title, and the and back piece is really my opportunity to, in part, give back to the community, and I spend quite a bit of time at the Salk Institute. I'm currently vice chairman of the board, and I also work with the UCSD Cancer Center and a bit with Pete Schultz over at Scripps. So it's been a absolute fantastic opportunity, and I've kind of been able to balance the community and focusing on drug discovery over the last few years. So if I start to drill into the Aragon and the Saragon story, I want to start a little with just cancer statistics. And cancer statistics really are that probably if you look around the room here, each and every one of you probably has some kind of personal story with regards to cancer, either a family or friend who's been afflicted with this disease. In my case, I got a phone call from my mother when I was a freshman in college, and my mother told me that she was diagnosed with metastatic breast cancer, which obviously we were very, very concerned about. The good news was she had surgery. She had a radical mastectomy in 1978, I believe it was. My grandfather was actually a radiologist in New York, and probably, again, you wouldn't do that, but he treated his own daughter, and she lived another 40 years cancer-free. So we had the pleasure of interacting for many, many years, post her metastatic breast cancer diagnosis. But as you can see, over 40,000 women and over 30,000 men will die from breast cancer and prostate cancer in the United States. So there's clearly a great need for us to develop new therapeutics targeting not only breast and prostate cancer, but other types of breast and other types of cancer. And since this is kind of a general lecture on drug hunting, I thought I might first shift and spend a few minutes a little on kind of the historical overview of how far we've come in the last 100+ years in the treatment of breast and prostate cancer. And I'd like to actually go back to the late 1890s for a few moments, which was kind of a transitional time for surgery. If you think about it, early surgery in the 1800s was bloodletting, no anesthesia, not really very antiseptic conditions. And in most cases, as you can imagine, it ended in a horrible situation. But in the late 1880s, 1890s, all of a sudden surgery changed, and it changed for two reasons. It changed once with the introduction of anesthetics. Cocaine was actually one of the first, and opiates were the second. Of course, ether and others came later. But that at least allowed for an improved situation with regards to surgical intervention. And also the second piece was really an approach for surgical removal of the diseased tissue or organ. And there was a very famous surgeon at Johns Hopkins Medical School named William Halsted, and he basically said, perhaps if I remove the tumor, and in this case he was interested in breast cancer, so I remove the tumor from the mammary gland, I might have a beneficial effect on the outcome. And in many cases, he had a very, very beneficial effect, but in some cases there was recurrent tumor. And often the recurrence of the tumor grew around the region of the surgery. So he hypothesized that perhaps the surgery wasn't complete with regards to removing the tumor. And he coined the term, I'm going to do a radical mastectomy. And he first removed the entire mammary gland with some success. But then all of a sudden he said, well, maybe more is better. So he removed the pectoral muscle. He removed the lymph glands. He removed part of the shoulder. He removed the neck. And so this radical mastectomy became, as he called it, a super radical mastectomy, and then an ultra radical mastectomy. And you can imagine horrible disfiguration and a lot of times tremendous complications with regards to surgery. And it was literally 100 years later where we took the needs of a careful clinical trial to show that a lumpectomy in almost all cases is as effective as radical mastectomy with regards to tumor and tumor recurrence. But it took 100 years, in part because of the dogma of the surgical community. But in the early 1900s, you started to think about, can you add on other therapies in addition to the surgery? That was at the dawn of the radiation age. William Röntgen had discovered X-rays, and he had demonstrated that if you irradiate certain tumors with X-rays, you kill the tumor a little faster than a normal tissue. So you can imagine surgery, add-on radiation, and then the third component, again, not high specificity with regards to the tumor, was the dawn of kind of cytotoxic chemotherapy. And of course, here in the School of Pharmacy, I probably don't have to go much into that, but you have to remember back in that time frame, it was observed that mustard gases, right, were able to basically kill normal and potentially some cancer cells. And so they started to make derivatives of mustard gases, derivatives from World War II, World War I, and now you had surgery, you had radiation, and you had cytotoxic chemotherapy. And literally for the next 60 or 70 years, you were basically balancing the killing of the tumor cell and killing of the normal cell, and that's why you have basically a narrow therapeutic index. And the big question that we're all faced with is, how do you know thy enemy, right? If you can selectively understand the molecular basis of the tumor, maybe you can target the tumor and keep the normal tissue. And a very, very important observation occurred really by urological surgeon named Charles Huggins, who is a professor at the University of Chicago. And the observation that he made is as follows. If you remove certain tissues and organs from an animal, you can have an effect on the physiology of the animal. And what did he do? He removed the testes from a male animal, and he saw, for example, involution and shrinking of the prostate gland. So he made a link between a natural hormone that was produced by the testes and the effect on a reproductive and organ. And what he also observed was that in a lot of the dogs that he were treating, studying this type of relationship between a hormone and ultimately a physiological event, some of those animals actually got prostate cancer. So he asked the question, if I remove the source of the hormone, again, the male reproductive hormone, testosterone or androgens, perhaps I can have a beneficial effect on prostate cancer. And that's exactly what he did. He surgically castrated the animals, and in fact, he had a remarkable benefit on the prostate cancer in the animals. And the same thing happens, of course, in women, but there, of course, you're removing the female reproductive source of estrogens. So you're removing the ovaries, and when you remove the ovaries, you have not only involution of the reproductive tissues, but in women that have breast cancer, you basically can have, in many cases, a very, very beneficial effect. So here's the link between a physiological hormone and basically the link between the cancer. And it's really the first defined molecular link between a hormone and a physiological event, as well as the outcome with regards to cancer. Now, surgical castration in men or women is probably not desired with regards to treating breast and prostate cancer. And so there was a need for basically understanding, is there a way that you can chemically castrate an individual and come up with a hormone that blocks the activity of estrogen in women or androgens in men, and perhaps that might be an effective way for the treatment of cancer. And this really links to some of the work that I did when I joined Ron's lab as a postdoctoral fellow. And Ron was the first individual with a few others to identify the target of those hormones. So in this case, this is the glucocorticoid receptor, or the receptor for cortisol. It has a characteristic DNA binding domain because it's a transcriptional regulator. And in the carboxy terminus of the receptor, it has a ligand binding domain. And it really works in a fairly simplified manner in which the hormone is basically able to freely diffuse into the cell. These are nuclear receptors. So when it binds to the hormone in an agonist state, it turns on or turns off transcriptional regulation. And that's really the understanding between the key, which is the hormone, and the lock, which is the receptor. Many people over the years have contributed to this family, this super family of nuclear receptors. There's 48 in the mammalian genome. The two that I'm going to focus my attention are the androgen receptor, which is the receptor for testosterone or androgens in men, and the estrogen receptor in the case of women. And the model that we focused on with regards to tumors is that probably about 80% of men that have prostate cancer are hormone-dependent prostate cancer, and this is therefore driven. Androgen is driving the tumor, the tumor is addicted to androgen. And so what's happening is that the androgen ligand is binding to its receptor. The receptor is binding to genes involved, for example, in cell proliferation and driving the tumor to grow. Same thing happens in women, of course, but now you're substituting estrogen for the ligand and the estrogen receptor for the estrogen as the ligand and estrogen receptor as the receptor, again, involved in estrogen-dependent breast cancer. There were a series of compounds in the 60s, 70s, and 80s that were designed. They were either involved as antihormones, meaning antagonists to these receptors. Drugs like tamoxifen is an anti-estrogen. Drugs like Casodex is an anti-androgen for the androgen receptor, and therefore quite effective. So if you're diagnosed with hormone-dependent breast cancer, for example, ER-positive breast cancer, you're often treated with an antihormone like tamoxifen, or you're treated with a drug that blocks the synthesis of that hormone, an aromatase inhibitor. And fortunately, for many, many years, the tumor is completely silent. But then all of a sudden, as I say, tumors are smarter than we are, and you become resistant or refractory. And so the big question a few years ago is, what's the molecular basis of why you become hormone refractory? And if you understand the basis of why you become hormone refractory, perhaps you can design drugs that, again, will interfere with androgen or estrogen signaling that would work in this hormone refractory state. And that's kind of where we started the story and how I got involved. As I mentioned, the former head of the NCI contacted me along with Charles Sawyers and Mike Jung, and they were interested in thinking about starting a company in the context of focused on prostate cancer. And as I started brainstorming with them, we kind of said, well, good news is we should develop drugs for the context of prostate cancer, but you have to be politically correct, of course, and if you're going to be politically correct, why you only treat men with regards to prostate cancer? You had to think about treating women, and of course, you're not treating prostate cancer, you're treating breast cancer. And so we set up a company called Aragon, and we focused on developing novel compounds that targeted both breast cancer, so altering estrogen signaling, and androgen altering for the treatment of prostate cancer. And that was the basis of the company. We basically got $8 million in 2009 to set up Aragon. And if you kind of read some of the venture capital pages and new startups of biotech today, $8 million isn't a lot of money compared to some of the very, very large raises of money that you hear about today. But in that case, you have to remember that in the late 2008, early 2009, there were some macroeconomic issues that we were faced with. And I kind of was thinking about, why do I even want to think about setting up a company kind of when the macroeconomics are kind of going sideways? My wife told me I was crazy. But we were able to raise $8 million in May of 2009, and that was kind of the birth of setting up Aragon. And what we were really trying to focus on is the observation that Charles Sawyers had made in the mid-2000s, in which you have let's call it a hormone-sensitive prostate cancer. So this would be a tumor that would respond to what I'll call the first-generation type of anti-androgens, drugs like Casodex. And Charles in his lab, made a very important observation that in men that had failed Casodex therapy, the androgen receptor is not mutated, it's not translocated, but it's just overexpressed in the context of what we refer to now as castration-resistant disease. And so the idea is, if you now know the molecular basis that the androgen receptor is overexpressed, can you create in vitro or in vivo animal models that will mimic the human disease and then interface biology and chemistry to come up with next generation compounds? And that was really the focus and the original basis of setting up the Aragon story. And so here, let me just kind of go through some of the early work just to make this observation. So in an early stage prostate cancer cell, this is a human cell, if you stimulate that cell with androgens, testosterone, the cells will proliferate. If you add a first-generation anti-androgen like Casodex, it has no activity on its own, but it's able to completely antagonize the activity of androgen or testosterone. But in the state where the receptor is now overexpressed, again, testosterone or androgen stimulates the tumors to grow. Casodex by itself, alone, actually has some agonist or partial agonist activity, so it's actually stimulating the tumor to grow here, even though it has no activity in this earlier state, and it can partially block the activity of testosterone, but again, there's some basal stimulatory activity with regards to the tumor, and that's why Casodex is actually in late-stage prostate cancer counterindicated, because it probably will stimulate the tumor to grow. So the question is, can we use this type of a model to come up with compounds that are pure antagonists? And this goes to some of the work that Mike Jung and his lab had done early in the lab working with Charles. Mike, as I mentioned, is a chemist at UCLA. He started with old Roussel-Claff compound. It was actually an AR agonist, and classic SAR, doing some structure-based drug design, as well as other types of normal SAR, identified regions of the molecule that could convert an agonist to an antagonist, others that were involved with regards to an affinity towards the androgen receptor, and then other types of chemical interactions that allowed Mike and the team to identify a compound called ARN-509, and you can see it binds to the receptor with very, very good affinity. I'll just point this out that when we started Aragon, we decided to license some technology from UCLA, and I'll come back to that a little later in my story. But typical of many startup companies, you license technology from academic centers, and then you build upon that as you build the company out. But it was an important license early on with regards to some composition of matter that we were involved with. How does that compound 509 and some 509 derivatives work? Remember, I told you that in the context of castration-resistant prostate cancer, Casodex has partial agonist activity. You can see over here, 509 is a pure antagonist, and it can completely antagonize the activity of a synthetic androgen. So it behaved, at least in vitro, as we were hoping. It's a pure antagonist, and it can completely block the activity of the natural hormone. And as we detailed additional types of mechanistic understandings, and so did Charles's lab, there were two interesting observations. The first observation is that in the absence of a ligand, the androgen receptor is normally in the cytoplasm. When it binds an agonist, it translocates to the nucleus, and then remember, it binds to DNA and regulates transcription. And of course, in this case, it's a pro-proliferative activation with testosterone or androgens. In the case of 509, what happens is it binds to the receptor, but it keeps the receptor primarily in the cytoplasm. But even the small amount of the receptor that gets into the cytoplasm has another additional benefit, and it can't bind to DNA. So if it can't bind to DNA, it's really not activating the receptor signaling pathway involved in proliferation. So we think this compound has really multiple points of activity. It functions as an antagonist, it keeps it in the cytoplasm, and it blocks the receptor from binding to DNA. And so the prediction would be, in models of castration-resistant prostate cancer, where Casodex would fail, this type of compound should have some beneficial activity. And that's exactly what happens. So what we're looking at here is an animal model of human late-stage prostate cancer. Each bar in the histogram here, in the waterfall plot, is individual growth of a tumor. So what you can see is in all cases, in the control animal, the tumors are growing at low dose, and then increasing the dose, you can see not only the tumor stops growing, but basically at mid to high levels of administration where the compound is still very, very safe, you're actually getting regression of the tumor. And if we did the same study with Casodex, the tumor, in fact, would still continue to grow. So we have a human model of late-stage prostate cancer, and this compound seems to cause regression of the tumor. The other good news is, does it have an anti-proliferative effect? And what you can see is that if you look at the proliferative index by looking at Ki-67, there's a decrease, in the tumor, in the proliferative index. And if you look at DNA replication and apoptosis, there's an increase in the apoptotic rate. So it looks like it's anti-proliferative and it's promoting programmed cell death. So we filed an investigational new drug application, an IND, with the FDA. We needed more money, so in, I believe it was 2010, we raised about $22 million, and we said to the FDA, we'd like to go into human clinical trials. The design of the study is really as follows. We started with a single dose of 509. We looked at the pharmacokinetics of the compound. We need to make sure, of course, the compound was safe. And if the compound was safe, we went into continuous daily dosing in patients, again, that had late-stage prostate cancer. And obviously, the goal of the phase 1 study was primarily to look at safety, primarily to look at pharmacokinetics, but we were hopeful that we were going to get a beneficial response. The other thing that we wanted to do was to ask, could we look at a marker, a pharmacodynamic marker, to say, is this compound hitting its target? And if it's hitting the target, could perhaps we have a beneficial effect? In a normal, healthy individual, PSA hopefully is very, very low. But in men that have late-stage prostate cancer where their prostate has actually been removed, PSA is a marker of metastasis. And typically men that have metastatic disease have high levels of PSA. And if there's a mechanism to shut down androgen signaling, their PSA levels should go down. So we had an opportunity to also monitor PSA in the phase 1 study. And then the final thing that we did was we did F18 labeled positron emission tomography. So you can imagine if you take F18 labeled androgen, it should bind to its receptor. And with PET imaging, you should see where the receptor is present. And if you treat with our compound, we should knock off the PET imaging agent and we should decrease the PET response. So that was the way that we built our phase 1 clinical trial. And the good news, I won't go into all the details, is these are the first set of about 20 men that had late-stage prostate cancer. They had failed all current therapies of hormonal therapy, and you're looking now at PSA levels. And what you can see is that the large majority of the men had a very, very robust response. In fact, about 80 to 85% of the men had a decrease in PSA levels. And of course, we were very, very encouraged because if you just look at the levels to decrease 50% of PSA, again, about 60 to 70% of the men had a robust PSA response. And the other good news is that the compound was very, very safe. And I don't think I have the PET imaging. I'll have that for the breast cancer story, but we also got a very, very robust response with regards to PET imaging. So we were super jazzed, and we said, okay, let's think about starting a broader clinical trial. And as I showed you in one of my earlier studies, you know, in early stage disease, you typically have surgery and first generation anti-hormonal therapy. And then as you start to fail that red zone here of what we call hormone refractory cancer, you can subdivide those patients into various aspects of prostate cancer. And we divided them into 3 different groups, and we said, let's basically test whether 509 works in the early stage of what we refer to as non-metastatic CRPC. It's really micromets, and then later stages of castration-resistant but metastatic disease. So that was the good news, but I'm going to divert for a few minutes and tell you again some of the trials and tribulations that one's faced with in the context of a company. And if you remember, I told you we licensed a composition of matter patent from UCLA. And after we treated about our first 6 or 7 patients, we raised approximately, I mentioned, another $22 million to launch some of the clinical trials here that I was starting to tell you about. I got a phone call from the head of tech transfer at UCLA, and a little small talk, and then he said, Rich, I have to tell you something that's of significant concern to UCLA. And that is that there's a multi-billion-dollar mid-cap biotech company that we had licensed some technology to a few years ago, and they claim that they have all rights to technology that came out of Mike Jung's lab, and therefore the technology that we had licensed from Mike's lab. And in fact, we're being sued by this mid-cap biotech company. You can imagine, I'm a first-time CEO, just raised some money, just got some great PSA data, and I close my door and I go, holy, you know what? So of course I immediately had to contact our investors, and I'm thinking, oh, they're going to throw me under the bus and they'll abandon us. I called Mike, who is our IP counsel at Wilson Sonsini. We called our general counsel, and we quickly did some analysis with UCLA, and we felt that we had done nothing wrong. And so we convened an emergency board meeting, and we basically had, are we going to bet the company and fight, or are we going to walk away and concede that perhaps this isn't worth fighting for? And we basically said we're going to fight. And so, of course, my Ph.D. at that point turned into a J.D. And we interestingly identified, through the help of some of the lawyers that we were working with, 5 high-end litigation firms, and we literally gave them 2 pieces of information. We gave them the license from UCLA, and we gave them the lawsuit that was filed against UCLA, and we said, we'll give you 24 hours, you come in and you tell us how you'd fight the lawsuit on our behalf. And so every evening, Monday evening, law firm 1, 2, 3, 4, 5 came in over the course of 5 days. And at the end of the week, we chose a high-end litigation firm out of New York. And as I said, we immersed ourselves in the lawsuit, and it was basically, a bet the company approach. And I'll come back to where we ended up with. But we had that great PSA data. We needed to raise more money. And so I started to go around and talk to venture capitalists. And of course, I show this type of data, and they go, okay, I'll write you a check, because this is amazing. 85% of your patients are responding. And then I said, oh yeah, by the way, there's one other thing I have to share with you. And I didn't know if I should tell them about the lawsuit first or the data first. And being a scientist, I told them about the data first. But by the way, I have to tell you about this lawsuit. And I'd say the traditional venture capitalists were sympathetic to the point of, Rich, great story, come back when the lawsuit's taken care of. We'll see you later. And you can imagine this put us in a very, very tricky situation. But we brainstormed amongst the investors, and we decided to go to what I'd call slightly untraditional life science investors. And we went to a group in New York called Topspin. Topspin was founded by a gentleman named Jim Simons. Jim Simons, many of you know, is credited with being the first quant. So back in the 1980s, he's a mathematician by training. He set up a company called Renaissance. And Renaissance now is a multi-billion-dollar company. Jim is a multi-billionaire, but Jim and his wife are also unbelievably philanthropic. You might have heard of the Simons Foundation. And just a few months ago, actually, Pradeep Khosla honored Jim and his wife for all the great philanthropy that they've given to both UCSD as also the general community. Jim heard the story, and he said, look, if my lawyers think that this is legit, I'm potentially interested. And Jim, in fact, invested in Aragon at a very, very tricky time. And we're ever indebted to Jim and his investment group, because the story played out pretty nicely. So what we decided to do was we raised some more money. Our current investors were very, very supportive. And as I mentioned, we went into these 3 subgroups of patients, hoping that we would see a long-term beneficial response. And again, I'll just summarize a little of the data. But in the first group, so that's the group that has what I'll refer to as non-metastatic CRPC, so basically micrometastasis. And these patients are very, very high risk for metastasis to bone. With it, typically, 25% of the patients will progress to metastatic bone disease within a year, and then unfortunately to more aggressive disease over time. And I'll just point out, in this non-metastatic group, we treated on the order of about 50 plus patients. This is some of the earlier data. But you can see after 12 weeks of treatment, 91% of the patients had a very, very beneficial response. and that is a response of decreasing the PSA levels by greater than 50%. And you can see that some of these men had an even greater response. And it was a sustainable response. So if you look after 24 weeks, again, 91% of the patients had a very, very beneficial response. If you look at a little later stage metastatic disease, also very, very similar. 80 to 85% of the men were seeing a long-term beneficial response with regards to our drug. And then in much later stage disease, the response was diminished, but there was in fact a response. So we were very...
Normal response is about 2 or below 2 for...
Yeah, exactly, yeah. And some of these patients had PSA levels probably in the thousands.
Wow.
And they were being decreased to almost normal levels. So we were very, very pleased by this. But then you're kind of in a blessing versus a challenge. And the challenge was as follows. Our androgen receptor program was going well. I'll shift moments. In a few moments, I'll talk about our breast cancer program. But I kind of sat down with our board, and I said, literally, we had treated a little over 100, about 125 patients, very, very good response. But to do a pivotal registration trial, so that's a trial in which you're basically comparing it to the standard of care, and then ultimately a phase 3 trial in which you're going to the FDA asking for permission for approval, we estimated that we would have to treat about 1,200 men. We'd have to treat them throughout the world. It had to be a national trial. And we estimated that that trial alone, from start to finish, would cost about $150 million. Because the breast cancer program was also going very, very well, we estimated that in the 2013 to 17 timeframe, we would need to raise on the order of about $300 million for the company. So I went to the board and I had a conversation with the board. Okay, here are the options. Option one is let's try to go public and gain access to other aspects of financing, an IPO, get into the public markets. Look for a partnership. Go to a big pharmaceutical company and see if we can do a partnership. Go to a company that had an interest in breast or prostate cancer and see if we could partner one of our programs. We could try, the third piece is to sell the company and see if we could get a return and a company that was committed to the programs that we were building and hopefully have a long-term beneficial effect for the patient. And the fourth idea that I had was what I call a buy-spinco model. Meaning, could we find a company that was primarily interested in our lead program, our prostate cancer program, and could I somehow convince them to give me back the breast cancer program? So buy the prostate cancer program and spin out into a new company the breast cancer program. That was kind of an aspirational goal. The board liked the idea. Of course, sometimes it's pretty hard to have those conversations with big pharma. But I was fortunate enough that over the years I had built a good relationship with Johnson & Johnson. Johnson & Johnson had a big commitment to prostate cancer. And I think they realized that small companies are small, they're nimble, they move fast. And the idea of buying us in total was interesting, but they realized that our breast cancer program was pretty early. There were other companies that just said, if we're going to buy you, we'll buy all of you, and we'll decide what we do with the people. But I was able, in fact, to negotiate that buy-spinco model. And clearly, J&J was very, very committed to the prostate cancer program. And we took Aragon, and I didn't mention this before, but one of the challenges that you're faced with in the biotech sector is, how do you come up with a name of a company? And our goal, I should have pointed out, was to say, is there a way to get rid of the receptor? Remember, bind to the receptor, kind of that super glue approach, but could you also build in a neutron bomb? Could you degrade the receptor at the same time? So Charles Sawyers, to his credit, came up with the name Aragon, which stood for androgen receptor gone. Get rid of the androgen receptor. So we were able to literally craft a deal in which there were 4 contracts. It was pretty darn complicated. The lawyers earned their money, but we also paid them a lot of money, in which J&J came in. They bought the entire company. So we closed the deal on a Friday. They paid us $650 million up front. to buy the company, and then there were $350 million in milestones based on regulatory approval. I had to invite the employees of the company in on a Friday and terminate all the employees. They kept me as the sole employee over the weekend, and I told my head of clinical, if there were any serious adverse events, don't call me up. I told my head of regulatory, if you get an email from the FDA, don't open it up. They kept me as the sole employee over the weekend, and then I invited everyone back on Monday to set up a new company. And we had to obviously get financing from the same investors. J&J was very, very cooperative. And then Mike, of course, he had to split all the IP into either Aragon or the name of the new company. We originally thought to call it Aragon with an E, but as you know, there's some kind of science fiction books and movies, and we kind of wanted to stay away from that. So we called it Selective Estrogen Receptor GON, Seragon. And basically, the majority of the people came back on Monday, and we started Seragon on Monday. J&J also made us change names, interestingly, probably to keep it clean. And we set up Seragon with the goal of the same group of investors, basically the same team, and to come up with compounds that activate and inhibit the estrogen receptor and had the added benefit of destroying the receptor, so basically causing proteasomal degradation. And so what you can see here is a cartoon of what we were trying to discover is we wanted compounds that bound to the receptor, caused misfolded protein, it became ubiquitinated, and then degraded by the proteasome. And we were able to do that using some structure-based approaches, but also some compounds from the literature to come up with compounds, again, that had that SERD-like activity, selective estrogen receptor degraders. And classic drug discovery, first tier of screens, second tier of screens, looking at proteasomal degradation, cell proliferation, conformational analysis, in vivo, in vitro analysis, and safety to come up with these class of compounds. So what you're looking at here is a human mammary cancer cell, and you're looking at the percent of the receptor present after our compounds are added. And you can see the compounds like 810 and 1917 are robust degraders of the receptor. We had independent chemical classes of compounds. We decided to move 2 classes of compounds forward. They were quite safe. And they also worked very, very well in animal models of, again, late-stage breast cancer. So what you're looking at here is tumors that are growing, individual tumors. So in a vehicle-treated animals, the tumors grow. Tamoxifen has a mixed effect. Fulvestrant, which is a degrader, but it has terrible pharmaceutical properties, has mixed activity. A drug that failed in some late-stage clinical trials for Lilly called arzoxafine had a beneficial effect, and our compounds also had a robust effect in these xenograft models. Now, again, we were interested in working in late-stage disease, so we created these models of tamoxifen resistant. So here's a tumor that grows, vehicle-treated animals. Here's a tumor that grows in the presence of tamoxifen. So originally, tamoxifen slows down its growth, and then it takes off. And here's our compound that works in the context of a long-term anti-tumor response. But most importantly, we were interested in these tamoxifen-resistant tumors, could you basically transplant these tumors, and would they respond to our compounds? And we were favorably pleased, I'll just go through it quickly, that in these tamoxifen-resistant tumors, they failed to grow in the presence of fulvestrant, arzoxafine, but basically the tumors were completely regressing in the presence of our compound. So we thought we had a very robust degrader. It worked in animal models of resistance, and we decided to move that compound into clinical development. So these patients had failed tamoxifen, had failed fulvestrant, they had failed chemotherapy, and we were then going to sites like Memorial Sloan Kettering, MGH, and Dana-Farber. We tried to get here at UCSD. It took a little longer, but hopefully their phase 1-2 unit has picked up a lot of steam over the last few years. This was in the 2013-2014 timeframe. And here's some of the data that we generated using PET imaging. So here you're using F18 labeled estrogen, and you're looking for where is the receptor expressed in a patient that has metastatic disease. And as you can see, unfortunately, this patient has extensive metastatic lesions in the entire skeletal muscle. The Abdominal cavity, of course, this is probably a little of the drug present, but you can see, again, in the pelvic area, in the skeletal area, massive metastatic lesions binding to the estrogen receptor. When we treat with our compound, I think you can see a very, very beneficial effect. Now, this is just showing you that the compound is engaging and blocking the activity of estrogen. It's not showing you it has an anti-tumor response, but it's obviously telling you that we've achieved a dose which is safe and is giving you a positive pharmacodynamic readout. But the good news is we actually got biopsies from the patients, and if you look at estrogen receptor positivity pre-treatment, you can see also there's some estrogen receptor positivity post-treatment, but it's less. And importantly, looking at a proliferative index, you can see a pretty robust decrease in a proliferative marker like Ki67. And in fact, when we exited the story, and I'll tell you how we exited the story, we were starting to see beneficial responses. Now, the interesting story occurred in that when we thought about the bi-spinco model I was telling you about at Aragon, we had talked to multiple pharmaceutical companies. And some of the companies that we spoke to after we started Saragon actually immediately came back to us and said, you know what, we really like your second program. In fact, we like your second program more than we like your first program. Are you potentially interested in doing some type of a deal, license, regional deal, partnership? And ultimately, it got to the point where Genentech and Roche said that we would like to work with you, and we'd like to work with you in the context of an acquisition. So literally, right around our first anniversary, after we had raised only $30 million, Genentech basically made a deal that we couldn't turn down. So they offered us $725 million up front and $1 billion in milestones. And the link was that we had to stick around for an additional 6 months and help in the transition between Saragon to Genentech and Roche. So obviously, we were hoping to advance these compounds further into the clinic, but there's always this tricky balance of finding a partner that has deep pockets. And in the case of the prostate cancer story, J&J was very committed to developing our compound. And in the case of Genentech and Roche, they convinced us and we agreed that they were very well equipped to advance compounds in the context of breast cancer. And kind of as an addendum, then I'll just finish up. The good news was we won what's called a motion for summary judgment. So we went through 2+ years of a tremendous fight for the lawsuit. We believed in ourselves. The lawyers helped us out. And ultimately, when it got to the judge, just before trial, literally a few weeks before trial, the judge said he reviewed 2 years plus of data, and he agreed with us that there was no basis of anything that UCLA had done that was incorrect, and we won what's called a motion for summary judgment. So we won on the lawsuit. And then the great news, in particular for the patient, is J&J had done just an amazing job post-acquisition. And on Valentine's Day of last year, the FDA approved ARN 509, which is called apalutamide, or now also called Erleada. And it is approved for men that have non-metastatic, so that micromets, castration-resistant prostate cancer. It was a 1,200 patient trial. And the good news is that the FDA worked with us and then J&J to look at what we call metastasis-free survival or death, and it decreased that by 72%. So men that were not treated with our compound progressed within 16 months in terms of metastasis. And when you were treated with apalutamide, you extended that process by over 2 years. And just to put it in perspective, sometimes you're getting drugs that are approved with literally a few weeks of benefit. In this case, it's over 2 years of benefit. And Rick Pazder, who we give tremendous credit to, who is the director of the FDA Oncology Center, said it had robust activity, and they worked incredibly closely with both Aragon and J&J to come up with these novel endpoints. So, you know, you hear a lot of bad things about the FDA. I can't say enough good things about how cooperative they were in the context of working with Aragon, and I know J&J was also very fortunate to work very, very closely with the FDA. And then if you fast forward about a year, in January of this year, Again, J&J told us they wanted to continue to push this compound into other types of subsets of prostate cancer. They conducted a trial called the Titan trial, 1,000 patients, and they showed actually not only an increase in progression-free survival, but also overall survival. So they're very, very committed, and it's having a wonderful impact for the patient. J&J predicts that this drug could see over 1 million patients over the course of the treatment paradigm. So I'd like to just finish with kind of where I spend a little of my time. I kind of made a personal decision post-Aragon and Saragon that perhaps I focus on what I'm passionate about, which is startup companies. I've always spent the majority of my biotech career on, I call it the sell side. So I've been involved and blessed with working with great academics, companies like Oric, which again is a company that I helped start with Charles Sawyers, focusing on overcoming resistance in cancer. Again, remember, tricky names. Some other oncology-based companies. Local company here that I started with Ron called Medicrin, which is focusing on fatty liver, NASH, neurodegenerative diseases. So you can see the theme here is, again, going back to treating chronic diseases, diseases of the aging population. And I mentioned Vividion, which I'm super excited about, using chemical proteomics. I never could convince my wife to let me go to the big pharma side, so I did want to be on the buy side. So I did go a little to the dark side in the last year, and I do a little work with venture capitalists in which I critique now new startups, but I only do it 2 or 3 days a week. A month. Whew. Careful. And I did want to spend a certain amount of my time, again, on the give back side, and I've been blessed to interact with some of the great academic centers here in the community. And as I think I mentioned in the intro, I'm vice chairman of the board at the Salk Institute, on the executive committee at the UCSD Cancer Center, and actually also work a little with Pete Schultz on some of his drug discovery programs. So in kind of finishing, what are some of the lessons learned? There's no substitute for great science. You know, if you're thinking about starting a company, I'm so proud of the team and teams that we've built over the years. You can't do great science without a great team. You do need to bet on yourself, and you have to have a lot of thick skin. You know, when you're begging for money, whether it's an R01 grant or asking for VCs, you're going to get turned down more than you're going to be accepted. Adversity, as I shared with you, does create greater tenacity and potentially greater upside. And I also think, you know, sometimes the VCs get a bad name, but in the case of Aragon, I think our investors could have literally walked away very, very quickly from us and just said, give us the money back. We had a great group of investors, and they stuck by us, and we found some novel types of investors, like I mentioned, the Topspin Group. And you need to think about creative ways for clinical trials and clinical trial designs, and our team did that. And I give, again, my team tremendous credit for doing that over the years. I've been fortunate to work, again, with great teams over the years, and I'm blessed that actually 4 drugs that I've worked on actually have been approved by the FDA, and multiple INDs. But I think we go into this really wanting to help the patient, whether you're doing academic research and translation research or ultimately at a biotech company. You know, our goal is to deliver a drug that's going to have a beneficial effect on the patient. And to me, that's one of the proudest things that the team and I are proud of over the last 20 plus years that I've been in the biotech side. And there's almost too many people to individually thank at this point. I think I've hopefully convinced you that it's the team, it's the team, it's the team. I've been blessed with great mentors and academics at UConn, University of Minnesota, the Salk Institute, but 3 people I have to really give a shout out to. Ron Evans, who was both a mentor and now a very close personal friend. Mike and Charles stuck by us. They went through a lot also with the lawsuit, but they've been fortunate to be involved in having a beneficial outcome for the patients. My biotech colleagues, I mentioned the investors, and there are a lot of times where I was either on the road, running around, so I have to thank my family and friends as well for kind of putting up with some of the craziness of, you know, of scientific research. But it's been a great road, and I've really had a great honor. You know, sometimes I I really don't view this as work. You know, it's just great to get up in the morning and try to have a beneficial effect for the patient. So hopefully I've given you a feel for drug hunting and my experience, and open to questions if there's a little time. But thank you.
Thank you so much. We do have time for some questions. I'm going to try to get the mic to whoever.
I was just interested if you could say a couple words. It was a beautiful story about how you're going to overcome resistance in cancer now. Because that's something I think about a lot.
Yeah. Well, I don't think there's a simple answer there. You know, what we try to do is develop models where we understand the molecular basis of the disease. And we try to start in part with, you know, now, if you think about when I kind of entered biotech, it was really the age of the mouse, and you're doing everything in the mouse. I think now with access to human samples, understanding the molecular basis of disease much more, it's kind of how do we use human samples and create animal models from true human disease, understand the molecular basis of resistance, and then target those nodal points of resistance.
So once again, Rich, congratulations for your recognition. The question which I have is about your second molecule, 810. If I noticed, it was very high dose, 100 mg per kg, compared to your previous molecule, and the concept is very similar. So do you know why do you need so much of dosing, high dosing?
Yeah, it really didn't have great pharmaceutical properties. So PK was poor. In fact, there were some GI disturbances. So if you fast forward, Genentech has taken multiple compounds forward that have improved the pharmaceutical properties.
So the lower dosing,
yeah.
Second generation molecule.
Yeah.
Thank you.
How did you identify and establish those relationships with venture capitalists? Like, what's that process like?
Well,
He's a first-year grad student.
yeah. So most of the time, you call, you email, and they don't even answer your call or your email. Sometimes you get in the door once and they show you the back door. So it's a lot of networking. And my suggestion is that if you decide to go into a biotech sector, work for a while with experienced teams, because those experienced teams, just like in academics, you know, you can call the chairman of the department, you can call the head of a venture capital group, and you know, at this stage of my career, fortunately, a lot of those people will answer my call. But if I tried to do that 20 years ago, you know, no. And so, obviously, it has to be great science, but you know, find teams that have done it before. You know, now that I'm doing a little, as I call it, the buy side, if you asked me early in my career, I'd say 80% science, 20% team. And now I wouldn't say the opposite, but you're betting on the jockey and the horse. You know, you're not just betting on the horse.
You started with some historical things, and I think it'd be interesting to get your perspective, because there was a cardiovascular, neurologic diseases and cancer, but it's only cancer that Big Pharma has stayed with, and the other ones, they've shied away. I mean, I get your opinion on that.
Yeah, I totally agree. And when you think about diseases of the aging, right, cardiovascular disease is number one, cancer is number two. But remember, cancer is, as you well know, it's a subset of many, many different diseases, and probably so is cardiovascular, Alzheimer's. I think the problem is, one, some of the diseases like Alzheimer's are very difficult to diagnose. They're probably polygenic in many cases, and we really just, we don't know how to tackle those diseases, and unfortunately, so many companies have failed. On the cardiovascular side, the FDA hasn't been as cooperative, say, as the oncology division, and so typically in cardiovascular disease, they require a morbidity and a mortality study. You know, so you're talking about not even 1,200 patients, you're talking about 10,000 patients. And, you know, that's been very, very challenging. Obviously, we know now there's a great link between statins and LDL and cardiovascular disease. But the FDA, if you said, I want to develop a drug for HDL and reverse cholesterol transport, they'll still make you do a 10,000 patient trial. So pharma has just totally shied away from that, and it's really horrible, because those are diseases that we really need to think hard about. Same in obesity, right? I mean, we have an obesity epidemic, throughout the world, no less than the United States. And to develop a drug in obesity, most pharma won't even go close to it. Yeah, and diabetes, absolutely.
And cancer's got a quicker endpoint
Yeah.
too, so you get your results unfortunately
Yeah,
faster.
and again, the FDA's been pretty forward-thinking in cancer about not even asking for survival.
Yeah.
They're willing to take progression-free survival, metastasis-free survival. And if you identify, for example, the local company called Ignita here that identified a point mutation in individuals, and you don't care the etiology of the cancer, but you have that point mutation, they have almost an 80 or 90% response rate. It's just remarkable. And now, people are starting to truly say for cancer, there are cures. The CAR T story, if you can figure out which patients are going to respond, they're cures. Remarkable.
I had a scientific question. How many fold is the AR count increased in these resistance, and also maybe if there's a similar number on the ER side?
Yeah, so AR is increased a few fold. It's not 10 or 100 fold, it's just a few fold in most men that have what we refer to as castration-resistant prostate cancer. But that in itself, is enough for Casodex not to work and other anti-androgens not to work. In ER, there's not a lot of evidence to suggest that ER is overexpressed. So why, the tumor becomes addicted to ER, we kind of still scratch our heads. I'd say in about 15% of patients that have failed drugs like tamoxifen or the aromatase inhibitors, there's actually point mutations in the estrogen receptor in the ligand binding domain, and that's probably why those patients fail.
Thank you.
So with the 509, what's the primary mechanisms of resistance then? Is it point mutation in the binding site?
Yeah. So we partly looked at that. Charles Sawyer's lab and others have looked at that. So we identified in humans that about 12 to 14% of the patients had a point mutation in the ligand binding domain when they became resistant to 509. But why the other 80% fail, we don't know. Some of the tumors become more of what we call a neuroendocrine phenotype, and maybe they're completely androgen unresponsive. But if you take those cells that are unresponsive to, say, 509, and you use siRNA to AR, they respond.
Yeah.
So there's some tonic AR, but we just haven't been able to figure out why.
Well, we probably should wrap up. I want to thank you all for the great questions, and let's thank Rich again for the wonderful talk.
Well, thank you.